So last day I went with the basics of very basics of transcription in prokaryotes, and I explained a bit about the central dogma. So now today I will actually go into details with the transcription in prokaryotes. Also, it will be a repeat class, for the video recording was not done properly well. So I'm sharing the slide. Are you people able to see transcription in prokaryotes lecture number 13?
Yes, sir.
আমি পড়াচ্ছি। হ্যাঁ, so I'll talk about transcription in prokaryotes. So one famous personality, Jim Rohn, said, education doesn't cease when you leave college or leave the university. Education is a lifetime process. So I am still learning, I am still understanding. This is the truth. This is a lifetime process. I like this quote, therefore I put it. তুমি এ না লিখলে আমি তোমাকে কোনো চকলেটের পরে দেব না। It's a lifetime process. So basically learning is a lifetime process. লিখতে হচ্ছে বলে এবিসিডি লিখছি, তুমি তো লিখলে না। So the aims of today's class is, first is understand the transcription process in prokaryotes. Then you have to understand the steps into transcribing a DNA template into an RNA, from how a DNA and RNA is formed. Then we have to see the gene structure of prokaryotes. Then you have to understand the promoter structure and the terminator structure. It is very important that you understand what a promoter is and what a terminator is. Then you have to understand how transcription starts and how transcription is terminated in prokaryotes. So before transcription starts, we have to understand what gets transcribed and what is the gene structure in prokaryotes. Rather, you can say what is the DNA structure in prokaryotes. This is what we have to study. Light আছে. Okay, so let us see the prokaryotic gene structure. So this is a part of a strand of DNA. Let's write part of a strand of DNA. আমি পড়াচ্ছি। হ্যাঁ, so this region is the promoter. This region is the terminator. And the region 5', this is the region 5' of the promoter. It is known as upstream sequence, and the one 3' of terminator, that is the downstream sequence. And this region is the place which is transcribed or RNA is formed. Messenger RNA is formed from this region. The promoter terminator is not transcribed. The promoter and terminator is not transcribed. It remains as DNA. Only the transcription starts from this point and it is known as the transcription initiation site. Transcription ends at this point. It is known as the transcription termination site. The 3' region of the terminator sequence is known as the downstream sequence. The genes are composed of 3 sequence regions. First is the promoter, RNA coding sequence, and terminator region. We have seen in the last picture the 3 parts. Promoter, RNA coding sequence, terminator. This part is the RNA coding. So first we will discuss about the promoter region. As we have seen, it is upstream of the sequence that codes for RNA. It is on this promoter region that the RNA polymerase binds. This promoter region, it gives information from where the DNA is going to be transcribed. It will dictate the transcription start site or initiation site. Start site. Then the RNA coding sequence. This is the region that will become the mRNA. Will be transcribed and become mRNA. And we have the terminator region. This region tells the RNA polymerase stop transcription. Now let us see the transcription process. This involves 3 steps. The first step is initiation, second step is elongation, third step is termination. So where does the transcription initiation take place? Is there any place in the whole genome? This is the whole genome of the bacteria. Say, from where does transcription start? So in the promoter region, if this is the place where the transcription starts. This is the transcription start site. -10 base pairs away. Understand that there is no zero base pair. This is the transcription start site. It is +1, and then immediately before that it is -1. There is no zero. The first transcribed, the first nucleotide over there is +1, and nucleotide before that is -1. There is no zero. Remember, there is no zero sequence. Okay, so the -10 sequence. So this is transcription starts from here. This green is the mRNA start region. This is the RNA polymerase. RNA polymerase has two parts: the main polymerase and the sigma factor. Right, and this sigma factor, the sigma factor binds to a region known as has got a sequence of TATAAT, TATAT. It is also known as the Prigno box or Tata box. This is if this A is the transcription start site, different mRNAs. The consensus is TATAT. These are different mRNAs. Of different genes. So, and now in the -35, there is this -35 box. It has got a TG rich sequence, TTA GACA sequence. This is also known as the -35 sequence. It is almost conserved. That means it may not be the same. -35 and -10 may not be the same exactly, but almost same. A consensus conserved sequence can be made. Clear about the structure of promoter? Yes, no?
Yes, sir. Yes, sir.
Okay, great. So now let's go with the transcription initiation. The sigma factor binds to the promoter sequence at the -10, -35. In the last picture you have seen, this is the -35, -10. The sigma factor has bound. The core enzyme binds to the sigma factor, but DNA is still closed. This is the promoter DNA sequence. It is still closed. This is the -35, say, -10. This is the sigma factor on top of the core polymerase. This is sigma. This is the core polymerase. DNA still closed means still in double-stranded coiled position. Double-stranded coiled position. And this type of complex is known as the closed promoter complex. Now the RNA holoenzyme or the complete enzyme. Holoenzyme means complete enzyme. This strand is, then it is converted into, it's opened. This region is unwound. It untwists the double strand of DNA. So once it is open, this is the open promoter complex. Once the DNA strand has opened, it is known as the open promoter complex. Open promoter complex. The RNA polymerase binds to the -10 sequence and placed in position to start transcribing. The minus sigma factor binds, so the RNA polymerase is in this position. This polymerase is now ready to transcribe. So once the transcription starts, the sigma factor is released. So unless the sigma factor is not released or from the polymerase cannot move. So sigma factor moves, removes, and the enzyme RNA polymerase goes on forward transcription. So in the transcription initiation, the RNA polymerase holoenzyme or the complete enzyme with sigma factor. This is RNA pol plus sigma, that is the holoenzyme. First recognizes the promoter at the -35 region. এখানে -35 region, this region, the RNA polymerase recognize over it and binds to the full promoter. It binds like this to the full promoter. This green region is the promoter. The green region is the promoter. This is now the closed promoter complex. The DNA is still coiled, therefore it is the closed promoter factor. And this is the sigma factor which is bound over here. This is the sigma factor. DNA is still coiled. DNA is still coiled, therefore closed promoter complex. As the initiation continues, RNA polymerase binds more tightly to the promoter in the -10 region. If this is the -10 region, this region, binding of polymerase becomes more strong. Binding of polymerase becomes more strong. DNA has unwound. Therefore, it is the open promoter complex. And the initiating nucleotide comes over here. Nucleotide triphosphate. This N can be A, it can be U, it can be C, it can be G. ATP, UTP, CTP, GTP. There is no T in RNA. And the RNA coding sequence starts from here. This is the transcription initiation. Now transcription elongation. The sigma factor will be removed. The sigma factor will be removed. And the RNA polymerase core enzyme, it moves along to transcribe the DNA sequence into a single-stranded RNA of the coding gene. When transcribing, the RNA polymerase interact with DNA sequence, forming transcription bubble. Now, in the next figure, we will see, okay, what a transcription bubble is. DNA double helix is reformed as the RNA polymerase moves forward. So, let's draw it. This is the promoter. This is the RNA coding region. RNA polymerase over here. This is the promoter region. Sigma released. Then this starts to move along. This is the promoter region. The RNA polymerase is bound over here. It's moving in this direction. This is known as a transcription bubble. Hannah, clear?
Yes, sir.
Yes, sir.
Okay. The DNA double helix is reformed as the RNA polymerase moves forward. So this is the promoter. This is the transcription bubble which is formed. So this DNA coils back. Helix is reformed. Few RNA nucleotide newly synthesized form an RNA DNA hybrid within RNA polymerase. So this is a DNA strand. The RNA polymerase, the transcription bubble is over here. This is the DNA. A small region, this part I'm drawing. This forms a RNA DNA hybrid. This is the RNA. And it's happened. This is the RNA polymerase. As transcription proceeds, single strand RNA gets out of the RNA polymerase. As the transcription is moving in this direction, the single strand RNA is coming out of the polymerase. This is the transcription bubble. This is the DNA. This is the pink one is the RNA polymerase. This red strand is the RNA, is the mRNA which is being formed. This is the RNA DNA hybrid region. Here DNA is unwinding in this region. Here DNA is rewinding back in this region. And this is the coding strand. This is the template strand. And NTPs are being added. And PP, pyropPPI, pyrophosphate is being released. This cleavage of high energy bond gives the energy for this reaction to take place. Now the question is, does RNA polymerase have proofreading activities? If there is any way by which RNA polymerase can check if it is can check correctness of nucleotide incorporation. It has a 3' to 5' proofreading capacity. 3' to 5' means the RNA is being synthesized from 5' to 3' direction. Once one nucleotide is added, it checks if the right nucleotide is incorporated. If it does not fit properly in the opposite complementary region, something else has come, some other NTP has come. Opposite of A, only U can fit. If someone else has come, then it checks whether it's problem. So what it does, it removes that one nucleotide and adds the correct one. Then what it can do, it can remove several nucleotides and add those several new ones. Next is the transcription termination. So termination, there are specific signals for the termination of transcription, the stop signals. One is the important is a Rho factor has been found in bacteria to be very important in prokaryotes. First is the Rho independent terminator. They are known as type 1 terminator. And the second is the Rho factor dependent terminator. They are known as type 2 terminator. So Rho independent terminator. So if you see these two sequences, this green sequence and this sequence, when transcription happens in the, this is the DNA with inverted repeat. This region is opposite to this, is complementary to this. The two green regions are complementary to each other. So this is transcribed. This sequence is just complementary to this sequence. Result, they form secondary structure, here stem loop structure. Once the stem loop structure is the RNA polymerase, once RNA polymerase encounters such a structure, the RNA is released from the RNA polymerase. So RNA, once the polymerase comes to the structure, it releases it, and the RNA is released back, and transcription stops. This is row independent. Hairpin loop, GC rich. Then there is a row dependent terminator, type 2 terminator. The terminator is a sequence rich in C and poor in G. Here there is no hairpin loop formed. The row protein, it moves along the RNA and comes to the C-rich sequence upstream of the termination site. Just before termination site, C-rich sequence is there. And row encounters this. Row is a helicase. And it unwinds the DNA-RNA hybrid which is formed. Hybrid, which is formed when this RNA polymerase is moving along the DNA. And it is released. Okay. So I finish my class over here. Many people have got questions?
No, sir.
Okay, then I stop this, and then I will tell you when I will take this next class. Tomorrow is on Thursday. Let's see, I will tell you. Okay.
Yes, sir.
Thank you, sir.
You're welcome.